Hello, everyone. Good afternoon. Thanks for coming to our Chacruna Community Forum. My name is Lígia Duque Plateiro. I am the Education Program Associate at Chacruna Institute. I'm joining this conversation from Rio de Janeiro, and I would like to know where you are joining us from. So if you can, please share it in the chat. Today, we'll have a very interesting conversation about the topic, the investigation of ayahuasca's genomes. We'll have this conversation among Francisco Prodocimi, who is associate professor and researcher at the Federal University of Rio de Janeiro, Brazil, and regional president of the Brazilian Society of Genetics, Regional East, and Alessandro de Mello Varani, who is a researcher at São Paulo State University, Unesp, and works with genomics and bioinformatics and microorganisms and plants. In this forum moderated by Anya Ermakova, who has a motley background and broad research interests combining nature conservation, ethnobotany, neuroscience, and psychiatry. This will be a 1 hour conversation among our speakers, and later we will have a half hour for the questions. If you have any question for the speakers throughout this forum, please feel free to put your questions on the Q&A button, which you can find near the chat. You can also upvote your favorite questions there. We encourage healthy and productive conversations and appreciate the participation of a diverse community. We ask that comments remain on topic and offered respectfully. If folks do not follow collegial rules and behave with disrespect, we will exclude them from participating in this event and delete their messages from the chat. Disrespectful behavior includes name calling, threats, hate speech, attacks toward people or identities, harassment, insults, and insulting profanity. Finally, when this event ends, we will find the recording at the same link. You can also find our previews and upcoming crowdcast forums on the Chacruna Crowdcast channel. Thank you so much for being with us today. So welcome, Enya, Francisco, and Alessandro. Welcome, Enya.
Thank you very much, Lydia, for the introduction. And I'm delighted to start this community forum. Good morning, afternoon, or evening, wherever you are in the world. And I'll pass the words to Alessandro and Francisco, who can tell us more about where they are, what they're doing, what is their background, and most importantly, what is their research.
Okay, Anya, thanks a lot for the invitation. Thanks everyone from the Chacruna Institute. I would like to thank Marcelo Leite. I would like to thank Bia Labati, Anya, of course, and Ligia and Ibra and all the Chacruna staff that allowed us to be here today. It's a great pleasure and a great honor for us to be here. And we'll be presenting just the initial steps that we have been researching on the ayahuasca plant genomes. And now present here, Alessandro, would you like to make your hello for the people?
Yes. Good afternoon, everybody. It's a great pleasure to be here, and it will be a good opportunity to talk about our projects here. And we prepared some material to share with you, and it will be a great, again, pleasure to discuss this project with all you guys. And Francisco, I think you can continue.
Okay, so I will start to make my presentation here. Ibra, can you put the presentation there? Okay, here we go. So yeah, so we'll be talking to you about our investigation on the ayahuasca's genomes. I'm from a professor. They asked us to make a brief introduction about ourselves. I am a bioinformatician, actually. My PhD was in bioinformatics. I am a biologist and a geneticist. I did master's in genetics. And my PhD was in bioinformatics. I lived in England, close to Cambridge in Hinxton, where I did my PhD in the Sanger Institute, one of the biggest genome centers in the world. A part of my PhD I did there. And I also worked in France, in the US, in Germany, in Mexico, and in Italy. But I'm professor here in Brazil, and I'm a professor here in Rio de Janeiro since 2011, when I founded the Genomics and Bioinformatics Laboratory, on which we would like to understand the amazing Brazilian biodiversity using the force of next generation sequencing technology. That's a new technology to sequence genomes. And apply an algorithm to understand the genomes of Brazilian fauna, of many organisms from Brazilian fauna and flora. And then we want to understand their metabolism, the genomics of those organisms. We want to understand their gene families, duplication of gene families, the relationship between the genotype, the DNA, and the phenotype, that it's the actual outcome of this genetic information. And we do this with data mining. We have published lots of papers in the last decade. And I've been working mainly with the genome of aves. We have a publication in Science and Nature, publishing hundreds of genomes of aves, together with American groups and Chinese groups. We did the parrot genome, and we work with partial genomes of ants and plants and lizards and fish and monkeys. And we are always trying to understand, to have a look on their evolutionary biology, and trying to understand molecularly how those phenotypes, how those organisms may work. Okay, it's a huge work, the genome analysis, and we have been working quite long with genomes of plants and animals from Brazilian biodiversity. Alessandro, I will ask you to introduce yourself. Could you do that?
Thank you, Francisco. Yes, a little bit different from Francisco. I did my PhD in biotechnology, but always working with bioinformatics. And one thing which is very important to say is that to work with genomics, to work in projects involving the generation of new genome sequence, it's very important to use the bioinformatics and bioinformatics tools. And since my PhD years ago, I'm working with genomics of bacteria. I started with bacterial genomes. And then from the last 10 years, I started to work with plant genomics. Because as Francisco said, here in Brazil, we have enormous biodiversity, and this biodiversity, it's largely unexplored in terms of genetics and genomics. We have a lot of very fascinating plants here in Brazil. And as I said, since almost 10 years ago, I moved from bacterial genomics to plant genomics, which are much more, I can say now, after my changing, the plant genomes are much more complex than the bacterial genomics in terms of organization. You can pass the slide. Thank you. And my main research line here in my group, I forgot to say, I'm located in Jaboticabal City in Brazil, São Paulo State. It's a very small, very tiny city located near Ribeirão Preto. It's a bigger city in the state, which is both located 300 kilometers far away from São Paulo, the capital. And in my group here in Jaboticabal, I'm also working with these new technologies to generate genome sequence and to develop new softwares to deal with this genome sequence generated by these technologies. And one of my main focus line of research is related with genomic instability, or the more known, when you see on the newspapers, the jumping genes, which are small fragments on the DNA which have the capacity to move across different parts of the same genome. These jump genes are very important because they are directly involved with the biodiversity, with the generation of biodiversity, in fact, sorry. And I'm working studying this kind of genes, and with more focus in the impact of these genes in the plant genomes. Regarding the plant genomics, I started working with carnivorous plant genomes, carnivorous plants, as you can see in the bottom right figure in green on this slide. It's a structure of one carnivorous plant. We did the genome sequence of this plant. The name of this plant is Utricularia reiniformis. And now I'm working with other plants from Brazilian biodiversity, like cupuaçu, which is the another one in the other side, in the slide, where you can see a little bit yellow. It is the cupuaçu fruit. Yes, thank you, Francisco. And now I'm working with other plants, and we start to work with ayahuasca plants, which are a very interesting topic because these plants are very special in terms of genomic composition. Francisco, I think I can pass to you now.
Okay, thank you, Alessandro. So the script of this lecture was developed by Anya. I would like to thank Anya for just giving us many questions that made our script for this presentation. And one of the first questions that Anya asked us was, why do you started to work with the genome of ayahuasca? And that's my question. My answer, actually. I started because I took ayahuasca for the first time. I was in the Machu Picchu in Peru, and I went there in May 2016, and I paid a shaman to present me, to introduce me to ayahuasca. I went to the Pisac, the city of Pisac, in the Sacred Valley of Incas, and I took for the first time ayahuasca. And it was a very, very transformative thing for me. I entered in this ceremony being a sort of atheist, and 4 hours later, I was a sort of a spiritualist person. And I have had difficulty to integrate my scientific experience with my own spiritual experience that I started to have at this day in May 2016. And then I went asking myself why I came into that experience, being who I am, being a bioinformatics guy working in genomics. Then I have this spiritual feeling of a mission, of a purpose. That's a very spiritual thing. And I enter in the ayahuasca research, guided by the plants, and with a feeling that they told me that they want their genome to be revealed, and I would be the person to help and work on this. And then I started the genome project in Brazil, and I invited a friend of mine, the population geneticist Simone Santos from the State University of Paraíba. She was a long friend of mine, and I invited her to participate in the project, and she had contacts with the Union of the Plants, the União do Vegetal, and we started the project in 2017. And she invited other fellow friends and collaborators to this project, mainly José Beethoven Barbosa, and we started this project, and we took like 2 or 3 years trying to purify the DNA to get the DNA of those plants without success. And at some point, my long friendship with Alessandro, that I know him as well from about 20 years ago, I invited him to come into the project, and I was very glad he gave us more than we need for completing this project. He had experience with plant genomes. He had experience with DNA extraction in plants, and when he came to the project actually started and developed with much more quickly. So thanks, Alessandro. I'm very happy to share this with you. And now he's a PI on the project as well, together with me. And this is our group. We made a collaboration with the Union of the Plants, União do Vegetal, and they allowed us to go to their church. We went to Serra da Cantareira, close to São Paulo, and we collected chacrona and mariri and the vine there to make the whole sequence of their genomes. So our project is a project of plant genomics, right? We not work actually with the ayahuasca tea, the beverage. Okay, the ayahuasca for us is an inspiration, a great inspiration to work on plant genomics. We are working in the genomes of the 2 plants that compose the ayahuasca tea, that are the sacred plants for the Amerindians, the chacruna and the mariri or jagubi. I'll talk a little bit about those plants. We send the samples to the University of Arizona in the United States, and we bought the service to sequence the genome there in this platform that's named the Pacific Bioscience Platform, that I will invite Alessandro to talk a little bit in a couple minutes. So the ayahuasca, as you know, is made of a decoction of 2 plants, the chacruna, that's from the species Psychotria viridis, and there are also other species from the Psychotria genus, but in general, the Amerindians and the religions, they use Psychotria viridis. That's a plant from the Rubiaceae family. That's the same family of the coffee Arabica. And we purified the DNA in high quality DNA and high molecular weight DNA, and this DNA was very concentrated and allowed us finally to sequence this genome. The question on chacruna, as you probably know, is that chacruna, there is this present this molecule that's the DMT, that's dimethyltryptamine. Okay, that's a tryptamine that is a neurotransmitter. It's produced endogenously in the human body, but just in field conditions like near-death experiences, and it's probably synthesized in the pineal gland in these near-death experiences. But so we produce ourselves DMT, and the plants also produce DMT. And there are other tryptamines, like the serotonin. The DMT is a sort of agonist, as an analogous of serotonin. Okay. and also melatonin, that the sleep hormone, okay? And the DMT is produced by the chacruna, and it's a highly psychedelic molecule, okay? So we want to learn from chacruna how this plant produces DMT. So this is one of the clear questions that can be approached in the genome analysis. So here we have the amino acid tryptophan. It's an amino acid. It's a very common molecule needed for producing all proteins that we have in our body. And the tryptophan can be decarboxylated. This part here, that's carboxylic acid, COOH, is removed, decarboxylated to produce a tryptamine molecule. And this tryptamine molecule is methylated in this N here, this nitrogen, and a CH3. It's added here and there. So it's methylated 2 times to produce DMT. And in the human body, the tryptophan is decarboxylated with this enzyme that's named amino acid decarboxylase. And in the plants, we know that there is an enzyme named tryptophan decarboxylate that removes this COOH, this carboxylic acid, to produce tryptamine. And then we still don't know which enzymes will methylate this tryptamine to produce DMT on plants, although we know that the human has a specific methylase. We are not sure still. So this is a question that we aim to answer. Which are the methylases that will methylate tryptamine in the plants, in the chacruna? So we will investigate this very specifically. What else can I say about this slide? And then the coffee presents 2 isoform of this enzyme, the tryptophan decarboxylase, that decarboxylate tryptophan into tryptamine. And probably we are expecting that chacruna will present the same enzymes or very close set of enzymes that decarboxylate the tryptophan to produce the tryptamine. And then we will investigate which methylases the plant is probably using, okay? We know that the DMT is therefore this psychedelic molecule. The effect of DMT was researched by Stephen Zara. And he named DMT as a psychose mimic molecule that would mimic psychosis, okay? And we know that's a serotonin-like molecule, binds serotonin receptor, mainly specific one, but we don't know exactly how it works. I mean, it's a very important psychedelic that provides the people they take it, the psychedelic experience, okay? And there are also many uses for biomedicine of DMT, mainly psychiatry, for headaches, for substance use disorders, and anxiety, depression, obsessive compulsive disorders. And this researcher, Ed Fretzka, he published many other biomedical analysis of DMT, evidencing that DMT is a psychedelic substance that helps in psychiatric disorders. Also in cancer, not only the DMT, but also the beta carbolines produced by the vine, the mariri. So we are sequencing this genomes using a very modern technology by the enterprise Pacific Biosciences. And Alessandro, would you like to sort of explain briefly how is the Pacific Bioscience technology?
Yeah, this is it's a very important part because all of this project to be possible, it's necessary to one way to access the DNA inside the plant, and to access the DNA inside the plants and to read the DNA inside the plants. And the biotechnology is evolving really, really fast during the last decade, during the last years. And one technology which emerged during the last years was the Pacific Biosciences. The main difference between the Pacific Biosciences technology in comparison to the others, which we already have, which was developed more than 10 years ago, is the capacity of this machine. What you're looking here on the slide is a big one. In fact, in the slide it looked like it's smaller, but in fact the size of this is almost one person with well, I would say in meters, 1.5 meters more or less, the weight of this machine. But the point is, this machine can read long segments of the DNA. That means that you can access the information in bigger parts and in high quality. And just to have one idea of the impact of this technology, it was considered the method of the year, I mean last year, 2022, in genomic sciences, because you can access the DNA in long parts of the DNA in high quality. And this high quality enable us to understand the genomic composition and the genes, and more important, can provide to us means to understand what Francisco said previously regarding the DMT molecule and the pathways involved in the transformation of the amino acids and the DMT. Using bioinformatic tools, we can investigate it, looking in this genomic sequence. Okay, I think I can pass to Francisco for the next one.
Okay. So the genome of Chacrona first surprised us, right, Alessandro? We were expecting to have a genome about 2 gigabases. 2 gigabases is 2 billion base pairs of DNA. But we did cytogenetic studies that found out that it was probably double the size, 4 gigabases, remembering that the human genome itself is 3.2 gigabases. So probably the genome of Chacrona is bigger than human genome by 30% at least. And then we sequenced using this technology of Pacific Bioscience long reads. We have 100,000 of genome of sequencing reads, and we are struggling to assemble this genome because the genome is big and you have to sequence in parts, and after make a sort of puzzle to find which part it's follow other parts, and we are still struggling with this assembling this genome. Alessandro told me yesterday that he was doing another assembly to try to get it better. But we have already a sort of good genome assembled. We have big scaffolds that are these pieces of genomes, as we call it. And most importantly, we have most of genes, more than 95% of the genes expected to be found in Chacrona. We have evidence that they are on our assembly, so we have probably a good coverage of the genome, and the genome is almost complete at now. We still need to analyze carefully this genome, but the structural part, the assembly of the genome is very advanced, and the genes are already, we have evidence that we have the genes found on this genome. We have published the first manuscript that just describes the organellar genomes. I mean, the plants, all the plants, they have two organelles, at least the mitochondria and the plastid, the chloroplast. And these small genomes, we just analyzed them, we assembled them, we annotate the genes on them, and they are very much like as we expected them to be, as a plant that it is. And we published this first report that was our kickoff of the genome work in the PJ journal last year. But then you may ask, what can you know doing the genome, right? What we can know, how this related to all things that are happening in psychedelic sciences now. To be truth, we don't know exactly what we can know, and that's the most fun adventure that we are entering. We don't know, it's an exploratory research. We are doing some sort of data mining approach, and we do comparative genomics, compare the genome of chacrona and mariri to other plants, and to find differences. Okay, what we know that we can do is to find the precise chromosome position of all genes and all non-regulatory elements. This is named the genome annotation. We'll find that chacrona has probably 11 chromosomes. This, we have a preliminary data suggesting that the plant has 11 chromosomes. And we'll find on each chromosome, that's a big piece of DNA, in which position each gene is placed. This is the genome is the molecular anatomy of a species, like describing the anatomy of chacrona, but in molecular terms. Okay, and then we'll find the precise set of proteins and the sequence of amino acids of all proteins that chacrona presents. Okay, so when doing comparative genomics, we can find that chacrona may have some duplication of gene families. Maybe chacrona presents something specific on the production of DMT that is unknown, maybe not. So we cannot know at this point, because the research is still ongoing. But we are mainly studying in chacrona the metabolism of DMT. This is our most important issue that we want to study in chacrona's genomes. But we also want to find what we call genome markers for population genetics research and population genetics studies. For this, we need a broader sample collection. Now we're sequencing one single plant, okay? But to do population genetics, we have to have dozens of plants collected in different localities, different places, and in the whole distribution of chacrona, the wild distribution, okay? And we are very interested in that, and we submitted a project that's it was a calling named Amazon Plus 10, and we asked 2 million reais, about $350,000, to go to Amazon and find many places and collect many samples and sequence those samples. But this project was not approved. So we are still looking for another place to fund us. Okay, so we are interested in knowing the biogeography of the sacred plants. Not only us, there are lots of groups also researching this. And if it's not us, it will be another person. But the genome that we'll make available will ease any group that wants to make this populational study, because these genome markers will be available after our work, okay? And we can find mostly hotspots of biodiversity. We can find where if we have lots of collected samples in the whole distribution of Chacrona plant, we can find the hotspots of biodiversity, and this can help for the conservation status of Chacrona, because we'll find the pool, the genetic pool, where it's richer, and we can protect more those areas on which the genetic pool is more diverse and richer. Okay, so it's an the genome is exploratory. It's a data mining approach, and we don't know exactly what we can find, but we are hoping for that we will find interesting stuff. And then I'll just talk a little bit about the mariri, the vine, the jagubi, that is the liana, right? That it's the other species that makes the tea, and it's from the species Banisteriopsis caapi, and there are at least also 5 species and different varieties of the mariri non. Mariri is a Malpighiaceae, from the Malpighiaceae family. That's the same family that is from the acerola fruit. And we also were capable to sequence high molecular weight DNA, to purify this high molecular weight DNA, and to sequence the genome using the same strategy with Pacific Bioscience long reads. And for mariri, the people from Union of the Plants, União do Vegetal, they have already a couple varieties that they know that they are different. And a recent study published by our fellow Saulo and Jaqueline and Tainá, our fellow researchers from IMPA in Manaus, they proved that they are actually different lineages from Tucunacá, that is this liana that there's no knots, right? And the Caupuri, that's another variety of the liana that has these knots here. These knots, they're simply can be seen, and they can separate and distinguish these different varieties by the presence of this knot. But this is not only the difference between them, because there's also Caupuri variety without these knots. And then we are still studying, not only us, but our other groups, other Brazilian groups, the diversity of the mariri. The mariri, we estimated the genome in 1.5 gigabases. That is like half of the human genome. And this is probably what we'll have, about 1.4, 1.3 to 1.4 gigabases of a genome, 1.4 billion base pairs, okay? And it's probably divided in 10 chromosomes, 10 big pieces of DNA as well, that we'll know very soon the complete sequence of those chromosomes, because this technique is amazing. The Pacific Bioscience, together with the genome assembly, will allow us to actually describe the complete molecular anatomy of the vine, the Banisteriopsis caapi varieties. And we are also doing what we call the studies of transcriptomes, because the transcriptomes are the expressed sequences, right? The genome is statics, and the genome is the same in a single organism. But the transcriptome change, the transcriptome of the root, the transcriptome of the leaf, the transcriptome of the flower. So we can study different transcriptomes and see which sort of genes are produced in each tissue. Although this first approach we are doing is just to help in the genome annotation. That's a very specific part of the bioinformatics work, on which we'll find the genes that are in the genome, because the genome, there are probably 90% of the genome that they are not genes, and these parts are not transcribed, and we still have to know exactly which are the parts transcribed. But we have the transcriptomes for the Chacrona, and we have transcriptomes for the 2 varieties of mariri, Okay, so we are doing also transcriptomics, that's sequencing the RNA and expressed genes. I'll keep going. Alessandro, would you like to say something?
I think it was okay, Francisco. You said everything necessary. We can finish the presentation, then pass to the audience to ask us if they have questions.
No,
right?
no, come on. I still have a couple of slides.
Okay. Nice.
So the Mariri presents these beta carbolines that are these enzymes that they degrade the tryptamine, the DMT, to produce non-psychedelic molecules. So the beta carbolines present in the Mariri, they inhibit the monoamino oxidase to produce, to transform the DMT in a non-psychedelic substance. And then in Mariri, we want to learn which are the enzymes that will produce these carbolines. So this is our aim with the genome of the Mariri, to find the enzymes that produce the beta carbolines. These are much more hidden enzymes that are harder to find than the ones in Chacrona. Okay, we work with other plants as well. And along this path, we are becoming neuroscientists as well, because at first we were just plain bioinformaticians, and we are doing lots of reads. I thank Bia Labate to editing all the books, Marcelo Leite, Draulio, and Siddhartha Ribeiro, that's leading researchers in the ayahuasca communities. Also listening also to the Amerindian shamans and psychedelic scientists. And we are still catching up with the literature in neuroscience to understand better how we can take profit of the genome information to enter and to help in the psychedelic studies. Okay, the psychedelic science in Brazil, I feel like it's still a taboo in Brazil. The researchers do not take seriously. Siddhartha Ribeiro always said that you are sort of risking your career if you start to work with psychedelics. But for lucky of us, we have Marcelo Leite in Folha de São Paulo journal. It's a big journal in Brazil that he's always sharing new research and trying to interview people and bringing knowledge of ayahuasca and psychedelics for the main public. Also, of course, here the Chacrona Institute, and we have the regulatory approvals, and we are searching always for the research grants to keep our study going. Anya asked us if we have infinite funding. This is amazing. But there's no infinite Earth. So the Gaia resources are limited. We don't have infinite funding, but if we have lots of funding, we would make interviews, books, movies, and humanitarian help for indigenous people here in Brazil. We are very inspired by indigenous people. I've been in a couple ceremonies of ayahuasca with indigenous people, and it's really amazing. It's the best setting you can have for an ayahuasca experience. If we have more funding, we would exactly collect the plant populations and study population genetics and ecology of those plants, mainly to learn how to please the plants, how to communicate with the plants, and how to cultivate them better. Okay, I would like to make a cultural center in honor of the sacred plants and in honor of the indigenous traditional knowledge that brought us this amazing substance, this powerful healing substance. And I would like to have classes of indigenous language and listen to the Amerindian stories and to help them if they will, as best as we can. I would like also to create a sort of research scientific center associated probably with a psychiatric center for aiding people with the use of sacred plants, shamanic rituals, and other psychedelics. Well, yeah, now I can just yeah, I'll just finish this choose light. Can I, Anya, choose light more, or it's finished now?
Yes, yes, of course. Please go ahead. We still have about 15 minutes, and
Okay,
then we have half an hour and questions from the audience after that.
Cool. I'm almost there. So the social aspects involved in the ayahuasca ceremonies, they are criticizing for people for a tourism approach into the Amerindian tribes. There's a controversy for paying for participating of those ceremonies, if one should pay or not for religious ceremony. I am very okay with this. I'm not sure. The Indians in general, the Amerindians, they like it if you go there and you pay them for having the religious ceremony. This is a very beautiful side of the culture, and sharing this with us, I think it's worth, it's proper, and it's okay. But I may be wrong. I want to listen from you, your thoughts on this. I have participated myself on paid ceremonies, and I think it's okay. We are a little bit worried if this become very popularized about the conservation of the plants. So mainly for the vine, the liana, the mariri, because the mariri is more difficult to be cultivated, as the chacruna is easier to cultivate, and I think there's less concern regarding all of this. There are also the questions about the institutionalized religions, like the UDV. That's a very beautiful religion. Also the Santo Daime, that they are sort of the guardians of the traditions here in Brazil. The UDV, they went to the US Supreme Court for the right to take ayahuasca in religious ceremony, and they won this fight. And the religious use of ayahuasca in the United States is allowed today because of UDV lawyers that were very well in this trial, and they allowed. Of course, it's clear that it's a religious. I don't believe myself in recreational use of ayahuasca. I think there are other psychedelics may be used in a recreational way, but ayahuasca is very spiritual, it's very religious, and I don't believe in a recreational use of ayahuasca in any case. Okay. Most of the institutionalized religions in Brazil, they are Christian institutions as well. They are syncretic, they are reincarnationist, and they mixed the Christianity with the Indian tradition. And I think it's very proper, very beautiful, and I like it as well. I have a profound respect for Jesus Christ, and also for the sun, the moon, and the snake, and all the symbols that come up when one use ayahuasca. And I think it's at this point, at least, it's okay. I think it be people should know more and have more respect, more honored these ancient traditions, both from the Amerindians and the institutionalized religions. Also, the ayahuasca ceremony, it's a ceremony of healing. Anywhere you take it, it's a ceremony of healing. So it heals your physical, mental, emotional, and spiritual problems, and it can help with alcoholism, depression, addictions, and trauma. I believe that I don't have the precise data. There are some data for each of specific questions. Okay, and now there are lots of literature that presents data showing that it's really effective. Okay, also the ayahuasca ceremony is a ceremony of cleaning and purging, that they call it the side effects. If you vomit, if you have diarrhea, and you have some sort of psychotic outbreaks, this is sort of common or usual, or not that usual, but it happens on a ayahuasca ceremony. And this is your cleaning. I don't believe that people, sometimes they ask me, oh, I want to make an ayahuasca that I will not vomit. I think this doesn't make sense, because the vomiting and all the purging that you have is a part of your healing. So if you take ayahuasca, it's for heal yourself, and this purges, they are healing. They are taking out of you some sort of bad things to you. So I don't believe that we can make any sort of ayahuasca or any sort of yeah. In this case, I'm talking about ayahuasca, not the other psychedelics that doesn't have this side effects. I think the side effects, they are important for your healing and for the healing process that ayahuasca brings. And also the Indians, the shamans will tell you your healing will be based on your own deserving. If you deserve to be healed, you'll be healed. And then we come more with the spiritual nature of this beverage. Some people has this feeling of final judgment when take ayahuasca, when you see your life films and you judge yourself. And when you judge yourself, you are the defendant, you are the defense, you are the prosecution, and you are the judge. And you have to encounter your self-forgiveness there. And of course, promise that you not do the things you did and you are healing on that moment, you won't do it anymore. So you encounter with your own shadow, you encounter with entities that according to biology, they are inside your own brain, according to some studies published. And you have to accept, honor, and keep your shadow controlled. And that's also a very important issue when a shaman is trained to be a shaman. A shaman is taken generally in the jungle with some ayahuasca, and the shaman takes only ayahuasca for, I don't know, lots of days. And the shaman, for one Indian to become a shaman, he has to pass through this experience of being by himself or by herself in the jungle, only with ayahuasca, and manage to control his brain and his sort of oniric experience, hallucinatory experience together. And then the person is prepared to be a shaman, according to what a couple shamans told me. So it's been a great pleasure to be here. I thank mainly Bia Labate, Ligia, Ania, and all the Chacrone staff. I thank also Marcelo Leite from Folha de São Paulo, the teacher plants and the traditional knowledge from Amerindian ancestry and the originary people, my own friends, Amerindian friends from the Fukaxó ethnicity, Cacique Tídio, Pajé Soiré, Tawanã, Ixale, mainly between many others from Fukaxó. Our research group, Professor Simone Silva, Professor Beethoven Barbosa, Professor Vitor Miranda, Dr. Saura, and Professor Ana Paula. I thank UDV, the Centro Espírita Beneficente União do Vegetal, mainly the people from the Menino Galante Center, Maria Alice, Conselheiro Gil, and all the collaborators. I thank also the angels, guardians, and the light beings, and we honor the challenges, the temptations, and the shadows that come up along our path. I'll thank all the collaborators and supported. Our work has been financed at this point mainly by FAPERJ, that's the funding agents from Rio, but now we have a funding also, a small funding from CNPq, PQ, and also FAPESP from Alessandro. And I thank also the owners of the copyright of the pictures used here. Thank you, Anya, and I come back to you. Thank you.
Wow, Francisco, Alessandro, this is a mind-blowing presentation and the scope of your research as well and your interest. Thank you very much for sharing your personal story and all your research. Like again and again, it's amazing how much research there is in Brazil on ayahuasca and various plants and biomedical research. So it really is world leading in this respect. And I'm also really amazed with your commitment for open science, for sharing your data, and also truly comprehensive thinking about the implications for biodiversity, for cultures, for indigenous people who use these plants as well. So it's very rare when in science we usually always specialize in one thing, know about one aspect, but here you present such a wide scope and wide interest. So it's really, really humbling to hear this.
Thank you.
Thank you.
And I have many questions. And we also welcome for the audience to put your questions in the Q&A, and then I will read out the questions as they come. But while we are waiting for questions from the audience, I have many questions. I don't even know what to start. So, Alessandro, you mentioned you're working on this jumping genes. Can you tell me, have you noticed any in the genomes that you studied in CAPI, or in Psychotria viridis?
Oh, yes. We already have some preliminary results regarding the Mariri genome and regarding the transposable elements, the jump gene composition and Mariri genome. And what we know at the moment is that at least half of the Mariri genome is composed by these jumping genes.
Oh, wow.
This apparently, in fact, when I say half, the people used to think, oh, it's a lot. Well, but in fact, in plants, it's a normal feature. Most of the plants have more than half or their genomes composed by these jump genes. And this is one of the reasons why I'm studying this kind of elements, because they represent a large amount of the genome. And if they represent a large amount, they probably represent and develop important things in the genome structure, or as Francisco said, the genome anatomy of the chromosomes. What these elements can do in the genome, making rearrangements. And during the rearrangements, they can transfer one gene located in one region of the genome to another region. And this movement sometimes may disrupt the function of the gene, or some other times may enhance the function of the gene. That's why they are so important. And that's why it's so important to track and to detect all these elements, and why I'm interested in these elements. And one question which you can try to answer in the near future is if some of the important genes in Mariri related with the metabolites involved in the ayahuasca tea preparation are derived for some kind of the movement guided by these elements, which will be very interesting in terms of basic science to describe it.
And my next question is about plant genomes in general, because they are often very large and you often get twice or 4 times or even more times the number of chromosomes. why is that, and why is it so different from animal genomes? And I don't know anything about bacterial genomes. Like, why is this the case? And what role does it have?
In fact, the plants are very diverse in terms of genomic size and number of chromosomes. The plants are very different compared to the birds or the other animals and other organisms. One thing which is very common in plant genomics is one event which we call whole genome duplication. This happens many times during the different lineages of different plants. The genome of the plant duplicated, and then passing the time, the genome can duplicate it again, and in between these duplication events, the plant lost some parts of the genome, duplicate again. This is a very common feature of plant genomics. They are always evolving, we can say that, very fast in terms of enlargement or the opposite of the genome size.
Shrinking.
Shrinking, yes. Thank you, Francisco. Yes.
And Alessandro, tell us about the hypothesis that chacrona is an allopolyploid.
Yes, exactly. Yes. Because when we start the project, before to start one genome project, we need to go to the PubMed or literature research to try to see if somebody did something related your plant genome, which you are intended to do the sequence. And one of the questions which we try to answer in the beginning is there out there any paper describing the genome size, the chromosome number? Is someone in the past did any kind of analysis regarding it? And we found that for chacrona, they expected, as Francisco said in the presentation, that the genome is about 1 or 2 billion base pairs. Okay, when we saw this result on the literature, we think, oh, it's okay, it's not so large, we can do it with our resources, let's do it. We start the sequence. And when we receive the data, with the data and using computer softwares, bioinformatic analysis, we can determine, looking the data, the expected genome size. And when we look at the results, we looked at the results told us that the genome size is more than a double of the expected size according to the previous literature. And doing some more analysis, we see that there's a lot of repetitions in the genome. When I say repetitions, I mean different chromosomes, equal chromosomes, homologous chromosomes, which are derived from these duplication events, what I said previously. And with this kind of evidence, we believe that chacrona, which we are doing the genome sequence, is a genome which passed through a recent event of duplication. That's why the genome is much more bigger than previous thought. Okay, and that means that you can continue, Francisco.
And we found out the most of genes, they are duplicated as well.
Yes,
that's
yes.
A big evidence that we have had this whole genome duplication in a recent lineage of chacrona, but also we might have taken a specific variety that this happened. We don't know, so we'll need to investigate further to really understand if this is conspicuous of the species or if it's just this lineage that we selected from the union of the plants that has this duplication. We are not sure at this moment, but we will investigate this along our research.
Yeah, there is a lot of work to look at plants from different parts of Brazil and also other countries where it grows as well, to see also geographic variation, to see how many there are. And as you mentioned, that there are lots of ethnovarieties or different morphological and probably genomic types of Kaapi wine, and it's possible you also find this with Chacrona as well. Have you heard much from talking to indigenous people, like other ethno varieties of Chacruna as well?
Well, I think it looks like there are a couple other species of Psychodria. I think they said about 5, or I'm not sure. I'm not sure. No, 5 is for the Kaapi, but at least 3 other Psychotria species that might be used once in a while. But it's I don't have too much information about that. Alessandro, do you know something else?
In fact, it's the same thing. Our collaborators from the north of Brazil, near the Amazon, from Acre State, they have this information in more details. But in our last talk, they said something that we have 4 or 5 different varieties of Chacrona. But the point is, in terms of genetics, we don't know almost nothing regarding these varieties. And we need money to go there, make one trip to there and pick these plants, bring to the lab, and do some experiments to verify the genetic difference. And then we can say that they are really different or not. The species,
and I guess
varieties, yes.
This would have implications for taxonomy, and that's where you figure
Exactly.
out whether they are species or they are varieties or other subspecies, and what is the difference between them, and also understand the difference in terms of maybe amount of DMT it has or other alkaloids, and what are they can be helpful for, and what is the difference between those different varieties.
Yeah, they used to say that these different varieties produce different DMT amounts, which are related to the experience with the people which use these different varieties. And more important, you said one thing very important. Exploring the biodiversity of these plants. It's very important for the taxonomists, and not only taxonomists, but also for conservation, because we don't know what we have out there. We need to know to protect first. First and most important thing.
Yes, absolutely. I cannot agree more, and particularly now with the psychedelics and ayahuasca becoming more and more popular all around the world, a lot of these plants are being taken, and we need to understand the conservation implications of this. And this leads me to another question about, you talked about how your research can elucidate different biosynthetic pathways and help to understand how plants produce DMT and beta-carbolines. But do you know why do plants produce DMT? What is the biological role for the plants to produce DMT?
I'm not sure. Do you know, Alessandro?
Yeah, in fact, this is we can say that this is a 1 million question, because the DMT is produced by plants, is produced by us in some circumstances, and why the plants produce it? Why we produce it? Different plants produce in different tissues, organs. For instance, the chacrona produce in the leaves, and the Jurema preta, which the name is Mimosa. I don't remember the species names.
Mimosa hostilis, I think.
Yeah, there is another name. The Mimosa produce the DMT on the roots. Okay.
Mimosa tenuiflora. That's the one.
Yeah. And it's a 1 million question. It's a very important question to be attacked by the science in the next years, and by us in this project. We need to propose one new hypothesis related with the production of DMT.
In general,
yeah. Okay. Go ahead, Chico.
no, okay. In general, they are related to some sort of neurotransmission and some sort of communication as well, but we don't know exactly the mechanisms on which they use and they use to communicate. It can also be used as a hormone that triggers the development of some parts of the plant. But these studies are very speculative still. So there's nothing as long as I know that is really known about why the plant produce the DMT. That's as far as I can reach.
Yeah, we don't even know why humans produce DMT. There are so many theories and speculations where it is a neurotransmitter or not, what physiological role it has, if any. We don't even know where and when we produce DMT. We know that there is definitely endogenous DMT, but whether it's related to stress or to certain circadian rhythms, we still don't know.
Oh, sure. Yeah, the circadian rhythm is also is out. There is lots of theories. For example, our collaborator in the Juremas genome, Draulio, Professor Draulio Araújo from the Institute of Brain Institute in Natal, he said that the Jurema produce different amount of DMT along the year, and maybe along the day as well. So it may be related to circadian rhythms as well. You're right. This is another hypothesis that there is evidence that they are produced differently. The DMT is produced differently along the year, and maybe along the day as well. But along the year, I think it's very well known. So it's related to the circadian rhythm as well. And also, if you look at another sort of tryptamine, that's melatonin. Melatonin is the hormone for the sleep hormone, right? So when it gets dark, the melatonin starts to be produced, and as more it's produced, you get sleepy. And so as melatonin is a sort of tryptamine, probably DMT may be also related or regulated by the circadian rhythms.
Yeah, that would make sense. And I would imagine that it has some sort of very important physiological role, because so many plants across so many different plant families do produce DMT all across the world. It's not just one or two plants, it's hundreds of plants probably. On all the continents, you have all the acacias that produce DMTs, all the reeds, grasses, even some sea sponges. So that would be a fascinating question to investigate.
Yeah, this is probably true, because if you look Chacrona and Jurema, they are from different families, right? And both produce DMT. That's a good evidence that the DMT can be produced from very different plants from different and distant families.
Mm-hmm. And you mentioned that you have completed also sequencing of mitochondrial genomes. So what information can this give in addition to the nuclear genome sequencing?
Well, the mitochondria is this organ, this organelle that in general is said to be the machine that produces energy in the cells, right? It produces ATP, and ATP is the molecule that cells use for having energy. But also ATP is the nucleotide A from DNA, and so it's produced for the nucleotide A of RNA to produce RNA as well. So it's very important. And also the mitochondria, both in plants and animals, there are lots of these genetic markers that are these genes that are used to make a molecular datation, a molecular phylogeny of genes and proteins, and also to study population genetics, because it evolves in a different rate from the nuclear genome. And describing the mitochondria can help people to make PCR markers, PCR, this amplification of DNA, that will help them to get more a nicer and clearer approach on the population genetics and evolutionary patterns on plant evolution. That's what I would say, Alessandro.
Yes, exactly. This is the most important part of this smaller genomes, the organelle genomes. We can use these organellar genomes to develop genetic markers to study the field, the diversity. Exactly, one of the first questions, first question which we start our chat here, because it's very important to understand the biodiversity of these plants to create one plan or manage the conservation. And it's very easy to work with these organellar genomes to develop genetic marks and bring to the lab, and use in the lab molecular tools to identify and classify the different individuals collected in the field.
Mm-hmm. Yeah, and I guess this could be used also to make some sort of DNA barcodes where you can identify where the plant came from, and for example, later on, sometime in the future, you can identify, okay, this is from a cultivated plant, this is has been poached from some locality where it was not supposed to, and you can trace the plants easier with that.
Yes.
Yes,
that's the point,
right.
to trace the plants and to trace exactly what are you looking in the field. That's the point, yeah.
Mm-hmm. And how long does it take to completely sequence and analyze the genome?
Well, it can take your whole life, if you like. The problem is we have 4 billion gigabase for Chacrona and 1.5 billion DNA nucleotides to analyze. And I think the genome work is like to make art. You don't finish a work of art. You just do whatever you can, and you deliver to the world. We can be our whole life working not only with the Chacrona and the Mariri genomes, but in every genome we sequence. The amount of data is so huge, and so unprecedented that it's difficult to analyze. In general, we take like 2 to 4 years for a given genome, depending on the size of your group, depending on how much people are working, depending on money, on grants, if you can hire people to work with you. But our group is very limited, very small, right, Alessandro? We have a couple 3 to 5 people that are actually analyzing the genome. And so it takes years because we have to make sense of this huge amount of data. And it's not anyone that can work on it. I and Alessandro, we are programmers. We have to develop mainly our algorithms and our bioinformatics scripts to be able to analyze properly the genomes. And so we never know exactly how many time it will take. The time is the time of preparing the manuscript. I mean, to having the data and being able to tell some story with the data and preparing the manuscript. But at least we need to assemble the genome with the reads and annotate the genome, find all the genes, and annotate the proteins, find all the proteins, annotate the processes, find all biochemical pathways and enzymes in each pathway, and do some comparative genomics. And this is huge work. And we are very few people. but we are very inspired, and we will advance, and we are aiming to deliver at least the Banisteriopsis scapi genome this year. Late this year, we aim to publish the Mariri, the vine, the liana genome, and the chacrona is a little more difficult because there's this problem of being a tetraploid, and we may need more sequencing, but we are unsure at this moment. Alessandro is trying another assembly these days, but yeah, it can take years. What would you say, Alessandro?
No, in fact, we are generating right now a little bit of more data to try to improve the chacrona genome assemble. But I would say, Chico, you was perfect regarding the question, how many years and how long it take to analyze one genome. But I need to say that when we look one genome sequence, when we are working with one genome sequence, we need to make questions, look in the genome. And you have very specific and exciting questions when we're looking for chacrona and Mariri genomes, because we know all the things involved with these sacred plants. And maybe we need to focus on these exciting questions regarding the DMT production and why the DMT is produced by the plants. Try to elaborate one hypothesis, looking in the genome sequence, could be one good way to go, right? But as you know, and as Francisco said, it's like art, and art, you never stop. You need to, someone need to tell you, oh, okay, it's very beautiful. You can stop and play.
That's it.
Thank you very much for all this enormous amounts of work that you are doing on that. And I know it's not like the human genome projects where there were billions of dollars and so many people working on it, and you have a genome that's much bigger than human genome, and it's just you with all your love and dedication to do it. And my final question for today is something that you mentioned in your presentation, that you're working on the paper about new mechanism of effect of psychedelics that's been informed by your genomic work. Can you give us hints? What is it?
I'm afraid that I won't enter into this subject because, you know, I know very little about neuroscience, and I am now trying to formulate a hypothesis. And let me see. The question is, we know that the DMT and the psychedelics in general, they sort of promote a rebooting of your synaptic, the synaptic, how can I say, the synapses that you make in your brain. So there are lots of experiments already proven that the use of psychedelics, they sort of relax some synaptic pathways, and they pop up new, because the DMT and the psychedelic experience, they promote neurogenesis, the creation of new neurons, and synaptogenesis, that the development of new synapses. Okay, so the question is, sometimes the psychiatric disorders, they happen because you are in some sort of synapses that they are always doing the same pathway, the same synaptic pathway from here to there, from here to there. And the psychedelics in general, they can relax the synaptic way, relax this way, and pop up other paths on which you can access old memories, old traumas, old questions in your life by another path, another synaptic path. So that's the theory. It's not my I did this theory. There is lots of evidence, but I have a different approach to tackle how this work. But I'm still very preliminary on my thoughts and my ideas. But that's the idea. Promoting this neurogenesis and this synaptogenesis, the psychedelics allow the access to old memories, to old traumas in different ways. So you can sort out from a trauma, accessing this trauma from other points of view, for other ways, for other synapses. So there are the default mode network, the DMN. That's the how your synapses are used to access some memory. And the psychedelics and the ayahuasca allow to access those memories from another way, another synaptic path. And that's what I will write about. I'm still collecting evidence, reading the literature, and I will start. I didn't even start to write about this, but it's a new mechanism that I want to approach. Probably not in the genome of ayahuasca, but in a theoretical article about psychedelics in general. But this is very speculative at this moment. But we know that they promote this rebooting the synapses, and creating new neurons. And this is about the way I want to approach. Do you know something about this, Anya?
I know a little bit about neurogenesis and neuroplasticity. Yeah, but I don't know if it's how specific it is to DMT, because usually all those theories, they applied to all psychedelics, and that they work through serotonin 2A receptors either. And the recent paper in Science that it could be even intracellular serotonin 2A receptors, and then they activate those cascades that result in various types of neuroplasticity. But I'm curious if maybe there is something specific about DMT that does it slightly differently, or
this I am unaware, and the idea and the theory is more general in that case. I don't want to make a specific case for DMT, but a general case for the use of psychedelics in that way. And I think they all work very similarly. And in that case, I think it would be very similar the way DMT works, and also psilocybin, mescaline, and even cannabis, probably the endocannabinoids and the cannabinoids. They may produce this neurogenesis, synaptogenesis, and allow different paths to access old traumas and old memories.
Mm-hmm. Yeah, I guess it's a project for the future to understand the well, first of all, to understand how psychedelics work in general, and then also whether DMT or 5-MeO-DMT or psilocin or LSD have their own particular features that work on maybe particular, maybe through different secondary messages, like through functional selectivity. There would be subtle differences between them. And with DMT, and as you were mentioning in ayahuasca, it's not just DMT, it's the synergy between DMT and beta-carbolines. And that could be a reason why it's like would be more therapeutic, as you were saying before the presentation. But I'm aware that we are almost out of time, so I will pass the word to Ligia and to you guys if you have any final words to say. And from me, I just want to say a huge thank you for sharing your knowledge and your work.
So thank you so much. Anya, Alessandro, and Francisco. And thank you so much for the audience. If you appreciate the work we do at Chacruna and would like to help, you can support us by becoming a Chacruna member through Patreon, which grants many benefits. You can find more information on our social media and website. Our next crowdcast will be on May 10th at 12 p.m. PST, and this forum is titled Support for Psychedelic-Induced Crisis, Lessons from Brazil. So thank you very much, everybody. See you soon in our